Hello everyone, welcome back to my channel. In this session, I will be explaining on the LAC OPERON concept, which is a very important concept in class 12 biology. So it mainly belongs to chapter number 6, molecular inheritance and variation. So before we begin the session, do not forget to like, share, comment and subscribe. So let us begin with the interesting session, my dear students. The LAC OPERON concept was elucidated by two people, Francois Jacob and Jake Monod. The LAC OPERON concept was studied in E. coli, which is an bacteria. In the LAC OPERON, the polycystonic structural gene is being regulated by promoter and regulatory genes. You have to remember, my dear students, it consists of only one regulator gene, that is I gene, and it consists of three structural genes, that is Z, Y, A. As I've told you, the structure of the LAC OPERON consists of one regulatory gene, that is I, and three structural genes, that is Z, Y, and A. The I gene codes for the repressor, which is an regulator protein, and repressor is being continuously synthesized by the I gene. So the repressor, which has been produced by I gene, will block the operator region. As I've told you, the structural genes are three, that is Z, Y, and A. Now, the Y gene codes for beta galactosidase, which is also called as beta gal. The beta galactosidase or beta gal will hydrolyze the disaccharide lactose into galactose and glucose. The Y gene codes for galactoside permease, which increases the permeability of a cell to the beta galactosides. The A gene encodes an enzyme called as trans-acetylase. It metabolizes the toxic thiogalactosides, which is also allowed to enter the cell with the help of the permease. Hence, all these three gene products are very important in the LAC OPERON for the metabolism of the lactose. As you can see on the screen, there is a LAC OPERON structure and you can find the word inducer. Right? In absence of inducer and in presence of inducer. Who is an inducer here? So, the inducer will be the lactose. So, lactose is an disaccharide sugar. My dear students, lactose acts as an substrate for the enzyme, that is beta galactosidase or beta gal, in switching off and on the LAC OPERON structure. Hence, lactose is the inducer of the OPERON. In the presence of the inducer, so here inducer is lactose. Lactose is an disaccharide sugar which can be hydrolyzed by an important enzyme, that is beta galactosidase or beta gal, into glucose and galactose. These are the single sugars, and these sugars can be taken by which organism? It is bacteria. So as you can see on the screen, there is an OPERON, right? P stands for promoter, I stands for inhibitor, O is operator, and ZYA are the structural genes. As you all know that I will synthesize repressor mRNA, which undergoes translation to produce repressor protein, and when the inducer or the lactose comes and binds the repressor protein, it undergoes a structural change or a conformational change which turns into an inactive repressor. Now what will happen? The inactive repressor cannot bind to the operator region. And in that period of time, the RNA polymerase can now bind to the promoter region. The RNA polymerase comes and binds to the promoter region. transcription takes place with certain enzymes along with the structural genes. The regulation of the LAC operon will be positive, or we call it as positive regulation or switching on with the help of the inducer, that is lactose. Now, in the case of absence of the inducer, there is no lactose present. What will happen? Okay, so the I gene, which is already going to produce repressor mRNA and then the repressor protein. So this repressor protein can go and bind to the operator region. So can the RNA polymerase come and bind here? No, because already the repressor protein is blocking the region, right? So repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. So there will be no enzymes liberated from these structural genes, because this is known to be switching off, or the regulation of the LAC operon is negative regulation, or we can also call it as switching off of the LAC operon.